And Michelle, I'll get them to type their questions into the chat, and then I can interject and collate them together.
Yep, that's fine. Okay, so genomics is obviously going to have an increasing impact in medicine across and in healthcare in general. Key areas that are going to be really important are genetic testing, risk, family history. So these areas have always been important. For me and the research I do, cancer characterization and precision medicine is a big area of development now, where we're just looking, not just looking at an individual's genetics, but we're also looking at the genetic characterization of a tumor to understand what could be the best treatment, or what would be a little bit, understand a bit more about the prognosis of that cancer. Another key area that's coming online, particularly in New Zealand and more so in Australia, is pharmacogenomics, where we're trying to design drugs and treatments that are more targeted to treat individuals without adverse side effects. So looking at someone's genetic makeup and making sure they get the right drug. What else has also creeped in is kind of like the mainstream entertainment genomics or genetics, which crosses over, I think, as well into medical practice, where you get the direct consumer tests, you get the lifestyle advice based on genetic tests. And these are kind of unavoidable, and that you will end up dealing with patients who have had a direct consumer test, and they will come to you saying that they feel that they're going to get a certain disease or what their risk is. So this is something also that we need to be aware of. And so we can't learn everything about genomics. So I think the key thing, and what I'm going to try and do in the next sessions I've got, is really just like focusing on what the essential skills are, and then what the resources are that you can use when you need to draw on these skills. So key things are obviously interpreting reports, knowing where to refer, be able to communicate genetic information, and then just also understanding the limitations of genomics. So genomics isn't the answer to everything. It's starting to help us to understand the pathology of a lot of conditions, but by no means is it conclusive. And I think that's the most important lesson probably. Why is genomics important to ophthalmology? Well, because genomic testing has improved rapidly over recent years in terms of turnaround times, in terms of accuracy of tests, it's much more useful to not just ophthalmology, but across all medical disciplines. Because a lot of conditions are very heterogeneous, the more we can understand about the molecular pathology, the more we can target treatments and understand more about prognosis of a particular condition, and also understand a bit more about the inheritance. Generally, as with most conditions, there are two broad groups. There's the rare conditions. So these will typically follow Mendelian patterns of inheritance, and they often present in childhood. And then you have the later onset conditions, which also are likely to have other factors that impact on their prognosis and development, and are more complex diseases will have some sort of environmental impact as well. But they will also have a strong genetic basis. So this is the kind of areas that I think are really important, and the things that I will touch on. These may all be areas that you're really familiar with, and if so, then you can just like zone out for the next 30 to 40 minutes. So what I'm going to go over is some key genetics. I'm going to talk about how genes work, how DNA works, and then talk about epigenetics. So epigenetics is really important. That's kind of how our genome interacts with our environment, and it's how gene expression works, and how lots of drugs that we prescribe are also working in terms of switching genes on and off to have an impact in terms of the healthy or unhealthy phenotype. We're going to go through typical and atypical inheritance, and then next week we're going to focus on taking a family history, and we'll do some population genetics as well. And then the other areas we may touch on, or we may just move straight to the cancer genomics in the last session, but also the precision medicine, pharmacogenomics, cancer genomics, and then talk a bit about CRISPR and gene editing, sort of the future for genomics. Okay, so we'll start at the very beginning. It's like one of my favorite pictures. That's a sperm and an egg. So a human cell, every cell of the body, every nucleated cell of the body has 46 chromosomes. So as humans, we have 23 pairs of homologous chromosomes. We've got 23 from mum and 23 from dad. So there's dad and there's mum. And we have 2 sex chromosomes. Males have 1 X chromosome, females obviously have 2 X chromosomes. So this means if there's a trait on the X chromosome, then it can be masked in females, but males don't have a mask to mask a faulty allele, so they would normally present such as color blindness. Human chromosomes are grouped and organized on the basis of size. The larger chromosomes are the first numbers, so 1 through 8, and then they get smaller as the numbers increase. And obviously the number of genes per chromosome is relative to the size of a chromosome. We call this the karyotype, and all somatic cells of an organism have the same karyotype. So every single one of your 65 trillion cells in your body will have exactly the same karyotype, will have exactly the same genetic information. And if we want to clone you, that's what we would clone you from, any single somatic cell of your body. The gametes obviously have half the number of chromosomes because that's what happens when you produce gametes. When we talk about somatic and germline mutations, this is a really important distinction. So a somatic mutation is something that you acquire in a particular cell line. So with a cancer, you can have somatic mutations. And these are mutations that will occur that drive, or cancer driver mutations that have occurred to drive the cancer. These aren't inherited. You can't pass them on to your children. So somatic mutations are something that is non-transmissible. Germline mutation means something that occurred in your sperm or your egg that you can pass on to your children. So if I go out and I lie in the sun and I go and get a whole load of mutations in my cells, these will be somatic mutations. And if I have kids, I won't pass them on to my kids. If I have a BRCA mutation I inherited from my mum, which means I've got an increased chance of breast cancer, then this is a germline mutation that I will pass on, or have 50-50 chance of passing on. The chromosomes have a P and Q. So in terms of when you describe an area where a gene is, P is the petite, the small arm of the chromosome, and then Q is the other arm of the chromosome. And there's numbers from top to bottom on a chromosome that gives you a specific geographic location on the chromosome where that particular gene of interest is, or a particular breakpoint or a translocation. So if it says like 1p21, it would be that the breakpoint would be around here. Or if there's a translocation, you get told where the two chromosomes are and where the fusion occurs. So this is a karyotype. This is a karyotype from, and we can tell some basic information at this macro level. So this is at the chromosomal level. So this is a trisomy. So in this case, the individual has 3 copies of chromosome 21. And this is a trisomy 21 female. So we know it's female because there's 2 X chromosomes. So a trisomy 21 female has Down syndrome. So in this individual, they don't have the 46 chromosomes, they have the 47 chromosomes. And this would have occurred due to an error in meiosis, whereby one of the parents would have contributed, typically the mother, would have contributed 24 chromosomes. They would have contributed 2 chromosome 21s. And when that egg got fertilized, that brings in the other chromosome. So there are a few chromosome abnormalities or trisomies that are compatible with live birth. These are there's one monosomy, which is Turner syndrome, and then there are trisomies like Down syndrome, Edwards syndrome, and Patau syndrome. It's a trisomy 21, 13, and 18. And there's also the sex chromosome aneuploidies, which are also compatible with live birth. And these include things like Klinefelter syndrome, where you have 2 X's and a Y chromosome. Other types of chromosomal abnormalities include structural abnormalities. So these are things such as Robertsonian translocation. So when we had the pictures of those chromosomes, some of them are acrocentric. So they're kind of bottom heavy and they don't have much information on the top part of the chromosome. And these chromosomes seem to be more susceptible to fusing with other chromosomes, and they can cause something called a translocation chromosome. So there's 2 chromosomes fused together. And what happens is, for the people who have a Robertsonian translocation, if all of the genomic material is there, effectively they are phenotypically normal. The problem is when they try and make gametes, because when the cells try to split up the chromosomes, they get confused over which chromosomes go where. And so the person ends up passing on abnormal numbers of gametes, which then can result in trisomies. Non-disjunction is whereby this is often increased with increased maternal age, and it's where the chromosomes don't separate properly. And again, one parent can contribute 2 copies of one chromosome, again, leading to trisomies. So a key thing with translocations is whether or not they're balanced or unbalanced. So with humans, basically, in a crude sense, you want all of the genomic information. You don't want any more and you don't want any less. So if you have a balanced translocation, it means you've essentially got all of the genomic information there, even if the chromosomes have translocated or fused together. An unbalanced translocation or an unbalanced chromosomal mutation would mean that there's some chromosomal material missing. The extent of a pathology typically will depend on the amount of chromosomal information missing, because if you think about a chromosome, There's so much DNA within a small band or even of a chromosome that if you have a visible deletion or a translocation, then you're likely to have had a number of genes that are impacted upon, which will then have an impact on some sort of pathology. So this slide is just illustrating a non-disjunction. This is one of the mechanisms whereby you can result in a pregnancy that has a trisomy. So in this case here, you've got an abnormal meiosis, where instead of one copy of each chromosome going into each of the gametes or each of the egg cells, in this case here, for some reason, these two little chromosomes have stuck together and have been passed on to one egg cell. And this one gets fertilized, you end up having a trisomy. And so this is typically, again, another karyotype of a trisomy 21. In this case, we've got a boy with trisomy 21, so we can see we've got the Y chromosome too. When I talked about the translocations, the acrocentric chromosomes are ones like here, like 14. So you can see like it's kind of bottom heavy, so it's little waist, the centromere is right near the top. And so these chromosomes are much more likely, the acrocentric ones, to fuse with these ones here, and this can result in a Robertsonian translocation. And so if someone has a balanced Robertsonian translocation between a chromosome such as 14 and 21, they've got all the chromosomal information, but then they will, when they pass that chromosome down, they will end up passing down two copies of chromosome 21, the translocated one and an additional one. So this is just illustrating what a Robertsonian translocation is. What happens is it breaks at the little top bit here, and there doesn't seem to be very much chromosomal information in the bit above at the top, and it appears to have no impact on the person who's the carrier. And then this will fuse with the chromosome 21, and then you've got this translocated chromosome. So here we've got an example of a translocated chromosome. So it's quite hard to see unless you're trained to look at karyotypes. But this is a normal chromosome 14, and this one here is a 14 with a translocated 21 on it. So this individual is balanced. They've got if their parent would have been balanced, they would have had this translocation chromosome, but this individual will have trisomy 21 because they've got two normal copies of 21, and then they've got this extra one that they inherited as a balanced translocated chromosome from their parent. Okay, so that's kind of a genomics at the macro level. So when we look at the karyotype level, we can look at the large scale. And then if we zoom in, we need to look at the molecular level, which is at the DNA. So really to understand about genes and gene expression epigenetics, we kind of need to understand what DNA is and what it does. And so I'm going to go in a quick 101 of where is DNA and what it does. So every cell of your body has these chromosomes. So I've explained what the chromosomes are, and all your chromosomes are in the nucleus. Every chromosome is made up of a long coil of DNA, and segments of this DNA comprise genes. So we have about 20,000 genes that are functioning, that create every single protein that makes us human, that makes us us, that maintains our body, that's providing antibodies, that pretty much does everything. Now, some of these genes are switched on all the time because we need to do certain functions like making cells, like metabolizing food, things like that all the time. Some of the other genes were only switched on during development, or were switched on or were working at a particular level when we were developing as an embryo. So there's a very exquisite mechanism in place that controls gene expression. So although we have in every cell of our body the instruction manual to make us us, all those genes aren't switched on at the same time in every single cell of our body, because that would be crazy. So genes are switched on depending on what you need to be produced in that cell. So in a liver cell, you would have different genes switched on than what you would have in a neuron, because they have different functions and they have different roles. So the gene here represents essentially like a library book in a library. It's got the information to make the protein that is functional. The gene doesn't make the protein unless it's switched on to do so. When a gene is switched on, then it goes through that process of transcription and translation, which then will produce the protein. So when our body needs a protein, a gene gets switched on, which is the instructions to be read to produce the protein. In transcription, the message gets read in the gene and gets converted from double-stranded DNA into single-stranded messenger RNA. This messenger mRNA then moves out of the nucleus and into the cytoplasm, where the cellular machinery, included like ribosomes, which is essentially the little protein factory, then read that piece of messenger RNA and produce this resulting protein. And then the protein can be packaged or modified and used and exported from the cell to be used as a functional protein. When we talk about pathologies relating to mutations in genes, you can see an impact in the DNA, or a mutation in the gene will have an impact then on the resulting protein. And it will either impact in resulting a trunk, either no protein being produced, which obviously is going to have a pathological effect, or you can have an aberrant protein being produced, or a protein of lesser function, or a non-functional protein. So any kind of mutation that occurs or variant that occurs will have, or likely to have, an impact on a protein. So if we look at this slide here, we can see that the DNA made up of the nucleotide bases, the 4 bases A, T, C, G, and it's coiled around these little structures called histones, which make a nucleosome. So essentially, this is like string winding around a bobbin to make up the structure of the chromosome. When the DNA is coiled up as a chromosome, you can see then that if there's a visible deletion or a big mutation, when you can see down the microscope, it's likely that that's going to knock out a lot of information, including multiple genes. This structure here, the histones and the coiling of the DNA, is really important in terms of how we understand about how genes get regulated and switched on. So if a DNA is tightly coiled around histone and compacted, it's less likely to be able to be switched on by other proteins that need to access the DNA to switch on the gene. This diagram here is just talking about transcription and translation again. Just to recap, in transcription, that's when you make single-stranded mRNA from your template, and it's only from the template of the gene. So not the whole massive billions of nucleotides that you've got, but just the template of the gene that you want to produce the protein from. And when the mRNA is produced, it's exported to the cytoplasm, and here the little ribosome, the protein factory, moves along, translating the mRNA, calling in amino acids, joining them together with peptide bonds and having this chain of amino acids. And the chain of amino acids essentially is what makes the structure of the protein, and it gets modified and exported. So we have a genetic code. The genetic code is essentially all our DNA is made up of. The coding part of our DNA codes for amino acids. So as I said, all proteins are made of amino acids. Our genetic code is relatively simple. We have 3 nucleotides, or a codon, that code for each amino acid. And each protein is made up of different sequence, a different combination of amino acids. There are two key amino acids, or key codes. ATG is, or AUG in RNA, codes for methionine. And methionine is always the start code in a piece when it's like a signal that tells the cellular machinery this is the start of the gene. And this is always the start of where a gene is transcribed and the start of where a gene is translated. And similarly, if we have a start point, you need a stop point. With transcription and translation, you have 3 stop codons. So the UAA, UGA, and UAG. So this is like a full stop in the genetic message. Once the cellular machinery gets to one of these stop codons, it tells the cell to stop transcribing to make mRNA, or in the case of translation, it tells it to stop bringing in more amino acids. That's the end of the protein. So this kind of, this information is important because this is how you can understand the impact of a mutation. So it's quite easy for a single nucleotide to be mutated through error in replication or whatever it is. And if nucleotide is mutated, obviously it can change the amino acid, or it could even change it into a stop codon, which is likely to have then a greater impact in terms of what the resulting protein is. So I'm going to move on to what the structure of a gene is. I thought I'd try and put it into context in terms of like a question or something. So this is a sort of a, if you've been referred, you've referred the parents of a 12-year-old boy for genetic counseling. He has features of type 1 neurofibromatosis, complicated with learning difficulties and motor deficit, manifesting with gross and fine incoordination. The NF1 variation testing confirmed an intragenic deletion involving exon 15 close to the upstream intron. So it's like a lot of it is just language and kind of like directions of where a gene is. But the bit highlighted in red, I think is kind of like an important thing just to see whether a testing point to see whether you understand what this means. So if you were given this descriptor, do you understand what an intragenic deletion is? Do you know, involving exon 15 close to an upstream intron? Would you be able to describe to someone what that meant in terms of just the geographic location of that mutation? So that's a useful thing to be able to do. So essentially, Intragenic obviously means it's within the gene, it's a deletion, and it involves exon 15. So in genes, we have introns and exons. The exons are the coding bits that make the mRNA. Exon 15 will be the 15th exon in that gene. The introns are the intervening sequence between the exons. These are the non-coding parts of the DNA. And close to the upstream intron. So upstream, so in DNA, we give it a geographic kind of location from 5' to 3', 5' being upstream, so to the left, so where the start is, and 3' being downstream towards the end of the message. So if something is upstream, it means before the start in the promoter region at the start of the gene, and downstream is more to the right, the other side, and the 3' end of a gene. So a gene kind of looks like this. So
Michelle, we've got a question come up in the chat there. Are exons/introns the same as breakpoints?
No. So you can have a breakpoint anywhere. My understanding is in a chromosome, you can have a chromosomal breakpoint. So intron and exon is just the structure of a gene where a gene is transcribed. It essentially is a breakpoint in terms of how the gene how it joins things together. So here we have the structure of the gene. This bit here is the promoter region. So the exon, every exon, so in here is like the little yellow bits. These are this is the coding part of the gene. And these are spread out over large areas of each chromosome. And the bit in between is the intron. There is something called splicing and a spliceosome, which is a cellular mechanism that cuts out or loops out all these purple bits and gets rid of them, kind of like an editing system, and joins together just the yellow bits. So the splicing occurs to produce the mature mRNA. Now, sometimes we can have a mutation, and the mutation can occur at a splice point. And so it can stop the intron being spliced at the right place. Or it can mean that some introns aren't spliced at all. So you can have a mutation that disrupts splicing, and that would obviously result in a pathology too. In terms of what's meant to happen when a gene is expressed is that when a gene is switched on from the promoter, the gene is transcribed, and the primary transcript is getting rid of these, firstly getting rid of these exons by splicing out the introns. So the mature piece of mRNA should only be made of exonic sequence, and the exon sequence, the mature mRNA, is what is translated into the protein, not all of the other stuff in between. The stuff in between used to be referred to as junk DNA. It's like long before we knew a bit about DNA expression and gene expression. It's likely that this information, or this DNA, all the bits intron sequence in between, is really important for binding of other proteins that help to regulate this gene to be expressed. So it isn't essentially junk DNA, but it's not part of what the actual gene that is expressed in terms of the mature mRNA. You obviously can have mutations anywhere in a gene. In terms of exons, they're obviously numbered. Different genes have different numbers of exons. This is the upstream end of a gene, this one here. This is downstream. And you also might refer to it as this being 5' and this being 3'. And this is the way in which we read, the direction in which we read essentially DNA. So this is our piece of DNA. So now I'm going to describe how genes work. So we know that they're going to be transcribed to make mRNA and to be transcribed to make a protein, but they're not switched on all the time. So if you look at the diagram on this side first, basically when a gene needs to be switched on, something has to bind to the promoter region. It's called a promoter region because it promotes the gene being transcribed. So typically what will happen is proteins will bind to this promoter region, or something will bind to this promoter region that will then start the transcription of the gene. That starts the whole process that the gene is going to be transcribed. So once the gene is transcribed, or once something is bound to this region, then the gene can be transcribed, transcription and translation occurs, and protein is produced. So this has a molecular homeostatic mechanism in terms of it can be self-regulating, but then this can also activate other proteins, or it can inhibit this pathway to control the amount of gene that is expressed, thus controlling the amount of protein that gets expressed. And this is again really important when we talk about things like epigenetics, and we talk about how environmental factors, how drugs, exposure to other elements can impact on gene expression. If this area here, this this promoter region gets blocked, then obviously this can be like a way in which we can control, the gene expression can be controlled and genes can be stopped from being switched on or stopped from being expressed. This diagram here is like a nice confusing one. This kind of goes in a lot more detail of everything that kind of like happens upstream. So here, if we say this is the promoter region of a piece of DNA, and this is the start of your gene, the ATG, the first exon of your gene would be here where it says coding region. This is showing you everything that kind of has to happen before that gene gets switched on. So there's a promoter region, there's some core sequence in it, and then there's all these proteins here, different proteins, transcription factors, activators, repressors, co-activators, all these proteins that act together and bend the DNA. So all this, there's a lot of this junk DNA upstream from a gene that is really important in binding these proteins to make sure that a gene gets switched on. So it's incredibly, I guess the point I'm making here is that genomics is incredibly complex. Genetics is way more complex than that kind of a binary relationship. We often think about genetics as being, oh, there's one gene mutation that causes one disease, that's it, there's one protein. There's a whole lot of other stuff going on. Humans are incredibly complex, and all of this kind of goes on to, in order to switch a gene on and to make the protein. So this is important. So again, this is the importance of gene regulation. So I like this, I really like this, this cartoon because it really, to me, illustrates the importance of understanding that not all genes are switched on in all cells. So here we've got a couple of eye cells talking to each other, and they're trying to read their instructions of what proteins they've got to make. And he's got the wrong instructions. He's been given the stomach cell instructions. And so the eye thinks it's got to make hydrochloric acid. And so in the eye cell department, they're making hydrochloric acid. And then they just realize that they've got the wrong instructions. They realize they've got the stomach cell instructions. And obviously that's going to go wrong because then the person's going to start making hydrochloric acid in their eyes. So this really illustrates, this is actually what happens in your body. You don't want your brain cells having the function of liver cells. You don't want your liver cells having the function of the brain. So different genes are switched on in different cell populations of your body. So this is an important concept to understand. If we're looking at gene expression and different proteins, you produce different proteins in different aspects of your body. So yes, your stomach would be making hydrochloric acid, but certainly not the cells in your eyes. And similarly, there'll be different proteins that are made in your eyes and different proteins made in your brain. So this is why epigenetics is really important. So the concept of epigenetics is where you have changes in gene expression without a change in gene sequence. So when we talk about mutation, we talk about either somatic or germline mutation. What we're changing fundamentally is the DNA. We're changing that sequence, and that mutation impacts on the protein. When we talk about epigenetics, we're not changing the DNA. We're not mutating the DNA or changing it. But what's happening is we're changing whether or not a gene gets switched on. So we're changing gene expression, the level of protein that's going to be produced. And this is a really important, as I said before, molecular homeostatic mechanism that regulates the activation and repression of genes. So it's really cool. It's a flexible mechanism, and it occurs during the development of stem cells. So this is why our cell populations were allowed to differentiate into different cell types. So this is why you have cells that look different. They've been allowed to differentiate into different cell types because different sequence of genes have been switched on or off that allow that cell to differentiate and become a specialized cell type. It's also flexible in response to environmental signals. So there's a lot of research looking at impact of diet and exercise on gene expression and how that relates to our health and physiology phenotype. When we talk about how this happens, like in terms of the molecular mechanisms, there are two broad kinds of epigenetic mechanisms that switch genes on and off. And these are methylation or histone modifications. So again, both of these mechanisms are referring to how easy it is to get a protein in to switch a gene on or off. So this slide here kind of illustrates a couple of the mechanisms that are really important in epigenetics. So here we have methylation. So methylation is a physical dropping or addition of a methyl group around that promoter region in the 5' upstream end of a gene. And when there are these methyl groups sitting on the promoter region, it's really hard to switch the gene on. They kind of block switching the gene on. So if a methyl group is there, often that gene is repressed. So And there's cellular mechanisms which can add a methyl group or remove a methyl group. And there are certain dietary components that are high in called methyl donors. So things like folate and betaine are high in methyl groups. So there's there are certain drugs that either strip methyl groups off or like they can add them on. So this is a mechanism whereby genes are regulated using via methyl groups by methylation. The other mechanism of gene regulation is on histones. So remember when I said that the DNA is coiled tightly around histones? The DNA, if it's coiled tightly, you can see here this little pink bit here. So if this is your gene and it's coiled tightly around a histone, it's really hard for a protein to get into this part and switch the gene on. So sometimes if a gene needs to be expressed, what happens is, rather than being tightly coiled around a histone, it gets relaxed and the histone expands like this so the DNA can be exposed. And when the DNA is accessible, then this means that the gene can be switched on. So these are mechanisms that are employed within our cells to regulate gene expression. So as I mentioned, this is really important for cell fate during differentiation and during our development. So here we've got two examples. So we've got the liver cell and we've got a neuron. So you can see here we've got the same gene here. So in the differentiation of this cell type into a liver cell, this gene here would be really important for development develop into a neuron will be tightly coiled, not exposed. This gene cannot be switched on, and another gene will be switched on, and this cell will differentiate into a liver cell. Alternatively, in a cell population that need to develop into neurons, this gene will be active and will need to be switched on because this will hold the instructions and the proteins necessary to enable this cell to differentiate into a neuron. So this is a really important mechanism where genes are employed relevant to what their job is going to be, what enable a cell type to differentiate into. And this is just showing the CPG island. A CPG island is just like a dense area where methyl groups are added. And typically we find these around the promoter region of genes that are repressed. So this kind of, why is epigenomics interesting and why has more relevance into research? Basically because it is the link between our environment and our genome. So there's all these environmental factors like toxins, diet, stress, exercise, smoking, alcohol, drugs, pathogens, even the weather, that will impact to a certain extent, either transiently or permanently on in terms of the genes that are expressed that will then impact on our change in phenotype, and whether that's changing physical shape, our disease susceptibility, response to stress, behavior, or longevity. This is a really cool experiment that really I think it really clearly illustrates the importance of epigenomics. So basically they took two mice and they were genetic clones. So they were the same genetically. They were both female mice and they exposed them both to BPA, so bisphenol A, which is like a toxin. And what happened was in one of the mouse mum, she had her babies, the bisphenol A stripped off methyl groups that were meant to be there, and a gene was expressed, which is called the agouti gene, that results in these little mice being yellow and fat. So she had babies that were yellow and fat with this rare gene being expressed. They did the same experiment with the other genetic clone of the mouse. And in this case, they poisoned her with bisphenol A, but they also gave her methyl supplements in her diet. So gave her lots of folate and betaine that then replaced those methyl groups that were being stripped off by the bisphenol A. And all her babies all resulted in being normal little mice. So although they were genetically the same, the dietary impact was counteracted by giving them the supplements, and her babies were all normal little brown mouse, whereas the other mouse mum, hers were all like yellow and agouti. So this is a really clear illustration that there is that environmental interaction with our genes that can change gene expression, which is obviously how we respond to the environment we're in. And this is really important.
Michelle, there's another question that's come up since we were talking about this. Is methylation also what happens with telomeres and aging? And I see you've got a pink aging arrow on that slide.
Telomeres, I think, telomeres shorten as we age. Methylation doesn't impact directly on the telomere length, but as we age, our epigenome does change. So if you start off with identical twins, they will have, there's a nice diagram, as they age, their epigenome becomes increasingly diverse, because an epigenome, so your imprint of is your exposure to everything. So whereas obviously their genome stays the same if they're identical twins, the epigenome becomes increasingly diverse from one another. As we age, our methylation patterns change. Methylation is associated with cancer, is associated with a lot of other diseases and pathology as well. I don't believe that it's specifically associated with telomere length, but telomere shortening obviously is associated with aging.
And there's another question, Michelle. So the genetic sequences that remain unwrapped from the histones within specialized cells, say our gastric parietal cells, do they remain unwrapped for the rest of our lives?
That's an interesting question. No, so I think because the cells have a limited lifespan, so the cells and then the DNA at that point will produce a protein, and I guess that protein will impact on those cells, and I guess that's a continuous process, but each cell will have and it's not just one cell that it's happening in, it's a whole population of cells, and those population of cells that live in that location. So that will be happening all the time, because that's what their function is, unless it was something that was important during embryonic development, in which case then they will, so genes that are switched on in embryonic development will then be closed afterwards. If it's something that we need constantly to produce cells or keep cells differentiated, then it will probably be retracting and unfolding. But if it's something that was only needed when we're in embryo, it would probably then permanently be folded up, because we won't need to access that again in that population. Is that okay? So in terms of, I mean, it's exciting, epigenetics, because it and a lot of the drugs that we prescribe them and they have an effect. We don't actually know, how we know that they work in patients, but we don't necessarily know how they work. And likely a lot of them is because they're controlling gene expression, or they're like blanket suppressing gene expression or something like that. So it's quite interesting in that whether or not we can reverse a lot of these modifications. So it's different to genetics. With genetics, we can't reverse the damage unless we do something like CRISPR and we stick in a nice healthy new gene. That's the only way we can reverse genetic problems. But with epigenetics, it's different, because it's not the actual sequence, that there is also that potential to reverse the imprint, or that the epigenome and what's happened to it by diet, drugs, or exercise, things like that. So, and again, it's kind of that relationship between, you tell your patients to live healthy and be healthy. The reason is because it has this molecular impact. So you're not just telling them to do it because it's good advice. You're telling them because it is actually doing something in terms of gene regulation. Yeah, this is kind of goes back to the nature nurture. So obviously twins, and when I started with genetics, twins were the go-to. So everybody wanted to study twins because they're like most genetically similar. But obviously now we're more interested in the impact of the environment as well. So a lot of the conditions that we face are conditions that aren't the rare genetic conditions. A lot of them will have an impact of environmental factors as well. And so this makes it more interesting in terms of understanding the epigenetics, because we want to try to understand more about how we can prevent or treat conditions based on the whole gene expression. So in terms of the history, genetics came from there's a whole push about Darwin and the survival of the fittest and evolution. And Lamarck was always laughed out of the evolution school because he said that giraffes got taller necks because they kept reaching for higher trees. And so this poor guy was told, like his science was rubbish, and then kind of like died a laughing stock. But essentially what we're doing is now with epigenetics is like we're kind of going back to actually he wasn't that wrong. Yes, the giraffes probably did evolve, in terms of what Darwin was saying, but actually in terms of epigenetics, we do respond to our environment. And if you're in a particular stressed environment or you're exposed to a lot of toxins, you will respond. Doesn't mean you're going to evolve as a species, but your body actually has this capacity to expand in terms of its just homeostatic response, a lot more than we probably gave ourselves credit for. And so that's why with complex diseases, we've got that whole multifactorial inheritance. And so again, I think it's a really good example when you look at something like heart disease. There's lots of genes, And so you can't just say like there's a gene for heart disease. There are lots of genes that are risk factors. And obviously some people will have an increased genetic risk because they have a family where there's increased genetic risk factors, and the same will be for certain other conditions. But there's also a huge amount of environmental factors, and it's really difficult to try and weigh up what the relative combination or contribution is to a condition that's complex in terms of what is hereditary and what is environmental. So we're looking at something like heart disease. Obviously you can have, you can inherit mutations in genes we know that impact on your metabolism, your personality type, whether you've got high blood pressure, the anatomy of your arteries, particular cellular mechanisms, your immunology, coagulation, blood type, and fibrinogen type. But equally, there's all of these environmental factors that arguably have equal impact in terms of risk. So in terms of your diet, whether you're stressed, you're ambitious, whether you get exercise, whether you're overweight, you smoke, your socioeconomic level, your education and culture. All of these arguably contribute just as much. So it's really important that we have a greater understanding of the complexity, I think, of genomics and epigenomics and how they interact. So I'll go back to a bit about mutations, because these probably come up more in your exams. So variations and mutations. So genes can have variations. So this is going back to the actual changes in the DNA sequence now. So this is not, the epigenetics is talking about how a gene works and how DNA works. Now we're talking about mutation, and we're talking about how genes can have a variation. So a variation can be benign, which is obviously not a bad thing. That means it's unlikely to have an impact, unlikely to change a protein, unlikely to cause pathology, or they can be disease causing. There are some mutations that are described as increasing risk, but not disease causing. So BRCA is increased risk of breast cancer. It's not disease causing, but it means you've got an increased risk of breast cancer. And then often variants get described as variants of unknown significance. And this is particularly important, particularly can be particularly important, I think, in New Zealand. We can have variants of unknown significance, particularly if you've got an indigenous population or populations whereby they may have a variation that occurs that's not documented in online literature or previous other literature. So you might send off someone's DNA to be sequenced, and it might come back and say there's a variant of unknown significance. This doesn't mean it's benign, and it doesn't mean that it's disease causing. It just means that it's unknown. So these are also important ones to be aware of. Benign variation can be used to track genetic information, but and mutations can occur throughout the genome. As I said, some have no effect, and some can produce quite severe disease. And often the size of a mutation is not a measure of severity. Obviously, if there's a great big chunk of a chromosome mutated, it's likely to have more of a severe impact because there are going to be a lot of genes there. But generally, in terms of the DNA scale, the size of a mutation isn't always a measure of the severity. So for example, sickle cell disease is a single point mutation that causes the pathology that results in sickle cell disease. So the important thing to understand is, going back to the DNA, what the value of that mutation is in terms of how it changes the DNA and how that DNA then is translated into a protein and how it impacts on the protein that is produced. The most common type of variation is a single base substitution. So these are SNPs. So when you do genome-wide association studies, when people are trying to map or make a relative association between a genetic mutation and a pathology, they often use SNPs. So these are single points of change that are common in populations, and they're used to kind of track or to identify areas of a genome that could be linked to a particular pathology. So going back to the DNA, we have here, again, some examples of some amino acids, and then the examples of the codons, the triplet that codes for each amino acid. And then we've just got some examples of some different mutations. So here we've got the amino acid lysine, and it's encoded by three A's. If there is a mutation that mutates one of these A's, adenines, into a guanine, it's okay because this codon also codes for lysine. So this would be a silent or a synonymous SNP, because you're not changing the amino acid. The nucleotide hasn't changed that codon, it's still the same codon, so it's unlikely to have a huge impact in the resulting protein because you've still got the same building block for that protein. Similarly here, we've got another one here, CAA, this one stays CAA. Then we've got aspartic acid. So in this case, we've got a codon that's made up of GAC for aspartic acid. And we can see here the other codon for aspartic acid is GAU. So if that occurred, it would be a synonymous one, but it hasn't. In this case, we've got a mutation where we've mutated the middle nucleotide. And in this case, the adenine is mutated into a guanine. Now, this case here, this mutation, a single change, point mutation of an adenine to a guanine, so an A to a G, changes this aspartic acid into now a glycine. So this is called a non-synonymous SNP. And in this case, we've changed the amino acid. So this could potentially have a greater impact in terms of resulting pathology because we've now changed the amino acid. And it really depends on the value of that amino acid in the total protein. If this amino acid didn't really, if it didn't change the charge of the protein or didn't change a binding site, it might be okay. But we now have changed an amino acid within that protein. And the last example here, we've got another mutation, which is called a nonsense mutation. And so here we've got lysine again, coded for by the 3 adenines. And in this case, the first adenine has been mutated into a thymine. And this has changed the AAA codon of lysine into now TAA. And TAA doesn't code for an amino acid. So in this case here, what you've done is you've introduced a stop codon. This can probably have, well, this will probably have a greater impact, particularly if this mutation occurs more upstream or closer to the start of that protein, because what you're doing is you're introducing a full stop or a stop codon at within that protein sequence. And so when that cellular machinery comes along and you're making mRNA and then you're translating that mRNA, it will stop at that point. So it will only string together the amino acids up until that point and then it will stop. So it will probably make a truncated protein. So a truncated protein is a shortened protein that is often unlikely to be functional. The other type of mutations that are important to know about are frameshift mutations. So this is where things get really complex. So if you imagine that this string of amino acids is your, this is your piece of DNA, and this is going to produce your protein. Your protein is made by stringing together these amino acids. If a frameshift mutation would be if you stick I have a mutation where you're sticking in another nucleotide or you're removing a nucleotide. So basically anything that isn't a multiple of 3. So you're sticking in a nucleotide here, because what happens is when you come along and you read it, it shifts everything down by 1 nucleotide. So it will change every single amino acid then. So a frameshift mutation can result from just the insertion of a nucleotide, but it can have a massive impact because it will then change every single amino acid that is read for, in terms of what the resulting protein is. And the other type of mutation that's important to know about is triplet repeat mutation. And this is where you have a codon, like in Huntington's disease or myotonic dystrophy, you have a codon that has a normal level of repeats. So in a normal person, it will have a certain number of repeats. So it's the same bit repeated over and over again, like a string of glycines or a string of lysines or something. And what happens is when this gets expanded too much, then it has a pathological impact, as in Huntington's disease. And so it will alter the structure of the protein because you've now got too many of these amino acids, whereas there should be a fixed number of them. This is just an analogy of types of gene mutations like reading. So chromosomes are like encyclopedias, genes are like pages of descriptions, and mutations are essentially misspelt words. And so in genetics, all our words are 3-letter words. So a missense mutation is just changing 1 letter or 1 word. So, you know, by mutating a particular nucleotide, you change the sense of what that sentence is. A nonsense mutation is that ending it too soon, so sticking in a full stop. So rather than having your full sentence, you've just got part of it. An insertion is this frameshift mutation where you're going to then change everything after the insertion. So you can see here we've got these two words are normal, and then you insert something and it changes everything. And similarly, a deletion mutation can have the same impact as an insertion mutation if you're having a frameshift impact in terms of the resulting mutations. So predicting the effect of a variant can be challenging, but in terms of like a traffic light system, generally it's likely to be bad if it's if a stop signal is lost or a stop signal is gained, if it's a frame shift, or if it disrupts splicing. So remember when I said you stick the exons together, you're splicing out the introns. So again, a frame shift and a splice disruptors can also have more impact in terms of what the resulting pathology is. Possibly damaging could be a non-synonymous mutation, so just changing a single amino acid, depending on what its value is, or an in-frame insertion or deletion. And likely not damaging would be mutations that occur outside of the coding sequence. So maybe 5' or 3' untranslated. So this is the bit to the left and to the right of the coding part of the gene. If it's a synonymous mutation, so the same amino acid, it's probably likely to be less of a problem. If it's intergenic, so within the gene, or intronic in the intron sequence, or in non-coding genes. And often if you are not sure, if you get a mutation, you can look to see what the impact is and see if it's an area of a gene that's conserved in other animals. If it's a mutation that's in an area that's conserved in other animals, it's probably likely to be really important, and so probably likely to have more of an impact. When we're looking at segregation to see whether or not a gene is disease dependent, so it's not enough just to identify a mutation. You've actually got to then have a plausible reason to say why it causes that disease. So is it making a protein that makes sense in terms of eye pathology, or if you just found a random gene and it's associating anyway. So you need your gene to be co-segregating with the disease in a family, and also ideally some prior association of that gene with the disease, and that it's in a pathway that makes sense. So often, like with SNPs and with identification of variants, they may be just close to a gene that could be interesting. They may not be in the gene itself that could be interesting in the pathology. So remember, there's lots of genes altogether, and genes are via linkage disequilibrium. Some genes are inherited together. So you may identify a SNP, but it may not make any sense in terms of the disease pathology, but it may just be that it's being inherited because of other genes that are close by. Likely benign variants can be, for example, genes that are common in a particular ethnic group and they're not associated with frequent disease. If, again, a synonymous change doesn't change amino acid, or a non-synonymous change of a non-conserved amino acid. The other important thing is also to know your population that you're testing, that different populations, different ethnic groups have different variants that are common and different variants that will be pathogenic. So there, again, is an important factor to recognize in terms of a population that you're studying. In terms of reports, the kind of the notation that's used. So you get the description of a change, and it gets preceded by a letter depending on what sequence is being used. So if it's genomic sequence, there's a G. So here it's like in genomic sequence that a position 76 and A is turned into a T. And C is for cDNA, so this is the exonic sequence. M is for mitochondrial sequence, R is for RNA sequence. And with RNA, you always have U instead of T, because RNA, you have thymine instead of, you have uracil instead of thymine. P is for a protein sequence. And so with proteins, each of those amino acids has a letter that represents it. You don't need to learn these kind of things, but if you see things with letters, know that that's amino acid. So whatever K stands for is the amino acid that's being changed to what an A amino acid is. Substitution shows you the less than, so greater than sign. So basically 76AC shows you that 76A is changed to a C. Deletions are designated by del. So 76 to line 78 del, or 76, 78 del ACT shows you there's a deletion of 3 nucleotides at this position between 76 and 78. And similarly, an insertion tells you that here an insertion is in T is inserted between nucleotides 76 and 77. So talking about amino acid changes, like I said, there's letters that represent each of the amino acids. So tryptophan is W and cysteine is C. So here it says that at position 26, so that the 26th amino acid in this protein, tryptophan was mutated and changed to a cysteine. And then you've got nonsense changes. If there's an X, it tells you that an X means a stop codon. So again, it tells you that there was a mutation, that there was an amino acid at position 16, which originally was tryptophan, and then there was a mutation. And so it's changed what it should be into a stop codon. So here's just an example using BRCA. So here you've got like BRCA1, you've got your chromosome, so it's BRCA1 17. It's telling you that the effect is genomic, in this case G to T, or in this case a deletion. It's telling you in the cDNA, then it's telling you what protein is affected, and then it's telling you the impact, whether it's non-synonymous or a stop signal gain. So typically this is what you would get in a report.
Michelle, there's a couple of questions that have been coming up on the chat here.
Yep. What Where do I find?
Yep.
Oh, in
So
chat. Yep.
the in frame insertion deletion. What does that mean?
In frame.
Yeah.
So within a reading frame, so your open reading frame is your exon sequence. So in frame deletion, but it means that it will be in a multiple of 3. So typically, so the reading frame, so because we read genetics in 3 letters, if you get a deletion that's 3 nucleotides, 6 nucleotides, or 9 nucleotides, it's not going to stuff up everything downstream. It's just going to remove or add in 3 new amino acids, or take 3 amino acids out. So it's an in frame. So it's in frame in terms it keeps the rest of the reading, the rest of your story or your genetic code is still the same. So all you're doing is adding in 3 extra nucleotides or removing them out. And so it could have an impact, but it's not going to stuff up as much as if you've got a frameshift mutation where it disrupts that sets of 3 and shifts everything along to become a different amino acid.
And the other question was, how come the stop signal didn't have an X in the previous slide?
Which one was that? It's probably me, I would imagine. So it's on the protein. So if it's on a protein, it has an X, and it's like the codon is given by, you can give the 3 codon as the 3 letters, as the nucleotide. Slide before this one. Okay. Oh yeah, so this here it's got termination, but yeah, you could have so TER is for termination signal, but you could have X as well. Yes, yeah. Is there other ones?
What are the numbers in front meaning?
These ones here?
I think the ones, the genomic ones.
Yeah, so these so this is the position in that. So so with genomics, genomic, when it's described, it's the position in the DNA. So essentially, it's massive. It's kind of like a library code, but it's where the whereabouts in the genomic DNA it is on chromosome 17. So where specifically on that chromosome that position is. So remember, like the chromosomes are massive in terms of the amount of DNA. So you're going to get a huge number for where it is in when it's genomic DNA.
And also, instead of writing W26C, can you write P26W greater than C?
Hang on. Instead of writing W26C,
yeah,
could you write W?
P26W greater than C.
Yeah. So no, you do P. You'd have it like this. So P. Yeah. Wouldn't have the greater than. You'd have P. If it's you would just have what the amino acid is either side. So you'd have you'd have it like this one here. You could write it like this, or you can write it like this. That would be the protein sequence, or you can just write it without. Yeah, the little symbol there is just pointing what it's going to. I think typically with the protein you don't have it. I think it's typically just with a nucleotide.
Okay.
Okay, so just kind of like going to move on to inheritance, because next week we're going to do family history. So now we've kind of done gene expression and we've done kind of mutations, but now we're going to talk about inheritance and kind of like paired alleles. So going back to the reason, that whole point that as humans we're diploid, we have 46 chromosomes, so our chromosomes are paired, we get one from each parent, so we have matched alleles. So if you have a mutation, you can have your matched allele is the same point on each chromosome, so you can have a healthy or wild type DNA, and you can have mutated DNA. You can get but it'd be at the same point in your chromosome from each parent, and that's called an allele. So an allele can code for the same or a different form of the trait, so depending if that's a mutation or not. If the 2 alleles code for the same trait, then they're homozygous. If they're different, then they're heterozygous. And so this leads us on to the sort of basic Mendelian inheritance that we get more associated with rare conditions, what we're going to play around with when we do family histories next week. So the types of inheritance, simple inheritance is like autosomal dominant and recessive. We have sex-linked inheritance, and then the more complex ones are the more atypical, where you have multiple alleles or incomplete dominance, or you have polygenic where there's multiple genes. With atypical inheritance, The key ones that will come up are imprinting. So this is interesting because this is where expression of a disease phenotype depends on where the mutation came from and becomes whether or not it came from the mother and father. So again, this relates back to that whole dosage. There are some genes where we only want a limited dose of the protein produced from that gene. And these genes are imprinted. So they are controlled so that we only get one dose. Particularly in the area of chromosome 15, and this is associated with Prader-Willi-Angelman syndrome, that the gene is switched off dependent on the parent of origin in which you inherited it. And so these conditions were discovered because individuals had these pathologies because they had effectively had no copies of functional or genes being expressed because it was silenced in one parent and then mutated in the other. So imprinting essentially is like, and it's done by methylation, so it goes back to epigenetics. So it's a way in which genes are controlled so that we get the right dose of genes. And this is really important for the X chromosome. So females have 2 X chromosomes. We can't have females producing double the amount of all of the stuff on the X chromosome because that's not how we work as humans. So a lot of X chromosome is inactivated, so it's suppressed or switched off on one of the X chromosomes of a female so that dosage is maintained. So females may have the same dosage of genes on the X chromosome as males because one of them is randomly inactivated or genes are switched off. So imprinting refers to that imprint that you inherit from a parent that enables us to control inherently some gene expression. And in some cases, there are conditions then that relate to this. I mentioned the other type of mutation. This is the triplet repeat mutation. So associated with things like Huntington's disease, Fragile X, whereby the length of a triplet repeat can determine a clinical manifestation of a condition. Mitochondrial disorders are interesting because they're inherited outside of your genome, so they won't follow a typical, a normal pattern of inheritance. So they'll be maternally inherited because we get all our mitochondria from our mums. You remember my very first slide, I had that giant egg and the tiny sperm. That giant egg has all your mitochondria in it. You don't get mitochondria really from your dad, you get them all from your mum. So if you have a maternally inherited condition, it's following a pattern of maternal inheritance, you could suspect that it's a mitochondrial condition such as Leber's hereditary optic neuropathy, myoclonic epilepsy, ragged red fiber. These are kind of, and generally the mitochondrial disorders affect tissues that have high metabolic energy requirements because it's the mitochondria that they're affecting, so brain and neuromuscular conditions. So these are interesting because we have a separate mitochondrial genome to our nuclear genome, so it's its own little thing. And then multifactorial diseases, I won't spend too much time on, but these are obviously most of the conditions that most people get sick from. There's multiple genes, environmental factors, and recurrence risks are increased if you have close relatives affected by an individual. And obviously the earlier age of presentation for most conditions, particularly cancers, generally indicates that there's a stronger genetic contribution. So I'm just going to go through some of the basic inheritance. So that was the atypical, now we'll do the typical inheritance. So dominant and recessive. These are phenotypes expressed by dominant and recessive conditions. So with a dominant trait, you just need one copy of that allele and you manifest that phenotype or that trait. With recessive, you need 2 copies. So with females, you have, so I was just looking at the question and getting distracted. I'll come back to that in a minute. So with recessive conditions, you've got 2 copies of the recessive allele. So in terms of conditions, albinism, absence of freckles in terms of kind of more normal phenotypes, attachment of earlobes, absence of dimples, flat feet, inability to roll your tongue, like that, they're all recessive traits. So they're all ones that you can work out whether you're recessive or not and what your genotype is. And there are other traits, including whether or not you can test things like certain tastes that you can test. Athletic phenotype is done on actinin 3. So there's lots of other phenotypes. You could probably have a guess at what your genotype is based on what you think your phenotype is. So this is obviously just illustrating recessive condition. So in albinism, the mother and the father are both carriers, so they have normal skin pigmentation, but that means that they can have a child that has recessive, inherits 2 copies of the recessive allele and then presents with the recessive condition. With sex-linked inheritance, so these are typically, we refer to genes on the X chromosome, although sex-linked inheritance could be Y chromosome. Y chromosome is pretty gene poor. There's not that many genes on the Y chromosome. So typically when we're talking about sex inheritance, we're mostly talking about the X chromosome. If a gene is only on the X chromosome, it's called an X-linked trait. And if it's not masked, it will be expressed in males. So color blindness and hemophilia are much more frequent in males because they're not masked. So color blindness here, mother and father, mother's a carrier, father's normal, and they can have female carrier children and colorblind males. So these are essentially just talking about, I find color blindness really interesting. So there's 3 opsin genes that control color blindness in humans, and they basically, this protein controls the absorption spectrum of light and determines the range of colors in which you can see. So obviously, depending on what your color vision is, you can either see the numbers or you can't see the numbers. And so kind of the variation, so genetic variation within the gene changes the protein, and the protein changes the absorption spectrum that you can see. So that's how it kind of relates in terms of the phenotype, in terms of vision. Interestingly, it kind of evolved through dichromatic. So this is like how a dichromatic and a monkey would see on the left-hand side, and then a trichromatic, we evolved color vision, monkeys evolved, and this is how they would see. And so it was an evolutionary advantage to have trichromatic vision because you can see predators more clearly and you can see food more clearly. So having trichromatic vision is an advantage, I think, that we've acquired. And different animals then see in different wavelengths, again, relating to mutations in these genes, which alter the spectrum of light that you can absorb, that you can see. So this is a really cool article. If you're interested in color vision, there's basically two chromosomes. We have chromosome 7 and the X chromosome, obviously associated mostly with color blindness. Red-green is on the X chromosome, and then you've got the blue-yellow on the chromosome 7. And so this is your protein that you produced. And so if you have these mutations, and mutations in the DNA can change amino acids. So here you can see these changes of amino acids, serine to alanine, or threonine to alanine, or tyrosine to phenylalanine. And these mutations, changing an amino acid, are sufficient to change the phenotype and changing the absorption of the light spectrum that you can see. So I think it's kind of just like a nice example of understanding the relationship between a mutation and the protein, and then what the phenotype is.
And Michelle, could I just interrupt you for a couple more questions?
Yes.
If mitochondrial diseases are supposed to be transmitted to males and females at equal chance, why do conditions like Leber's hereditary optic neuropathy affect males more than females?
I'm not sure.
So Paul has answered. Leber's hereditary optic neuropathy has the estrogen has a protective effect, so it's less manifested. So I guess it's about penetrance, isn't it?
About penetrance, and I guess maybe the level of mitochondria in different cells and muscle development, how males' bodies develop, muscle mass, I don't know, tissue mass, density of mitochondria, potentially. That's a good question, but really they're definitely all from the mum.
Yeah. What does recessive not masked mean?
So recessive, so if it's on an autosome, like on chromosome, any of the chromosomes, it's recessive, and it's masked because you've got one good copy. So you've got your recessive allele and you need 2 copies to get the disease, and it's masked if you have a good copy because the recessive one won't be expressed. But with X-linked conditions, men will it could be recessive because it's still a recessive trait, but they don't have a backup copy because they haven't only got one X chromosome, so it's not masked. So they've got the recessive allele and they've got no other copy, so they will get the condition. Whereas in females, color blindness is recessive, they need 2 copies to get color blindness.
But it's not masked by the good gene.
Yeah.
Okay. I don't think there's any other current questions.
Okay, multiple alleles. This is where you've got more than 2 alleles. So blood groups are a good example. You've got 3 alleles, A, B, and I, or A, B, and O. And so A and B being co-dominant and O being recessive. So there are other genes that you can have more than 2 alleles, and there's not one, so one isn't dominant, one isn't recessive. In this case, O is always recessive, but A and B are co-dominant. A can't beat B and B can't beat A. So if you inherit an A and a B, you are blood group AB, not A or B. Incomplete dominance, where you have an intermediate phenotype. Hair texture is a good example. Flowers, there's loads of them. So where you have a heterozygote that has a different phenotype. So you'd have, in terms of hair texture, you've got wavy hair as opposed to having curly or straight hair. And then lots of colors in plants where you have an intermediate phenotype that's not the red or not the white, it's in between. And so with polygenic inheritance, there are a lot of traits that require multiple gene pairs. So for example, and this is like just a very basic illustration of how we get the whole spectrum of different skin colors. So you can have, say if you had just 3 genes, so 3 genes that control skin pigmentation, there's 3 gene pairs, each with 2 alleles, then you can see how you can get a very continuous then variation in terms of the phenotype. So if you have in the first generation someone with very light skin mating with someone who's very dark skin, in the first generation you'll get, everyone will be obviously be heterozygote, but as generations go on, then you get a much more variety then of in terms of that continuum of skin color. So it just kind of illustrates with any trait that has multiple alleles, you're going to get a huge, more continuous variation or spectrum of phenotype. And same with eye color as well. I mean, although we say this is like brown and blue eyes, it's also a spectrum with eye color. And so here we have blue eyes is obviously recessive to brown eyes, and green eyes are also recessive as well. So in terms of a dominant and a recessive allele, the dominant alleles are the brown eye allele, and the recessive allele is the blue eye allele, and also the green eye allele. So the last thing I want to talk about is really just preparing for next week. So these are things we can just go over that make when we do pedigree analysis next week. So these are the different types of inheritance, and these are some key features to recognize. When you have autosomal dominant inheritance, you get vertical transmission through the generations, male and females equally affected, and an offspring has 1 in 2 for an affected parent. So if I've got Huntington's, there's a 50-50 chance every child I have will have Huntington's. Also, autosomal dominant conditions can be complicated by reduced penetrance and variable expression, whereby the phenotype may not fully manifest, or it may be milder or stronger depending on the genetic background that disease is presenting in. In terms of autosomal recessive, typically you'd only get, if you've got siblings, only one of them will probably be affected. Males and females, again, equally effective. And another red flag would be if there's any consanguinity in the family. So that's kind of any type of cousin-cousin marriage, or any other type of consanguinity, because obviously then you're limiting the gene pool. And the risk is 1 in 4 for carrier parents, and then a low risk for the affected parents. So again, this is where carrier testing is done for things like cystic fibrosis, Tay-Sachs and other conditions, whereby you can't tell if someone's a carrier because they're phenotypically okay. So it's important to know whether or not they've got an increased risk for having a child with an affected condition. X-linked dominant is less common. So this would be females to sons and daughters, males only to daughters, no male-male transmission, both sexes affected, and girls would be typically more than boys because some conditions would be lethal in males because it is a dominant condition. We don't see many of these. X-linked recessive, only males affected. Offspring risk for a carrier female are 1 in 2 for an affected son and for a carrier daughter. Offspring risk for an affected male is that all daughters are carriers, and you can't get male-to-male transmission. So the key thing to remember is that if you're a boy, you didn't get your X from your dad, you got your Y from your dad. So that's always the case. So it's kind of like just a thing to remember. A mitochondrial, only transmitted by females, offspring risk up to 100%, dependent on homoplasmy, heteroplasmy, so depending on the proportion of mitochondria that have the mutation as well. And both sexes are usually equally affected, but obviously, as you said, with labors, then the males are more affected. And then you have chromosomal inheritance. So this is where, when we talked about the balanced rearrangement, you can pass on this translocated chromosome. So each generation would have, so this translocated chromosome will stay within the family. And so these types of families would have increased risk of miscarriage because, you know, obviously the errors associated with meiosis. So again, if you see there's people in different generations affected, it's probably not going to be recessive. If you only see Affected members in one SIBSHIP, probably going to be recessive. If there's consanguinity, probably recessive. Only males, probably X-linked. More females than males, X-linked dominant, but very rare. Male to male, probably autosomal dominant, can't be X-linked. And males have no affected descendants that could be mitochondrial. These are just some questions, but we don't have to do these. You can do them if you want. So these are things that you can just practice for next week. So male to male transmission, whether or not it says mitochondrial, so it's false, that would be autosomal dominant. Parental consanguinity, autosomal recessive would be true. More
so Michelle, should we set these as homework? You've put your presentation up, haven't you?
Yeah,
but the answers
probably the answers as well, but it's probably worth just, we can just go over these. You can just try, and if you can work these out in your head, then you probably know you're probably fine for working out family history stuff. So yeah, maybe just go through these at home. I think that was all I was going to do. And then next week, this is what we'll do. We're looking at family trees. So we'll use these symbols, but we can go through this. So you can have a look at this at home if you have time. Make sure you're aware of like the symbols that we use, like the circles and the squares, and what we do when someone's dead, or if they're affected, or if they're a proband. Again, this is in the presentation, and we'll refer to this again next week as well. I think that's all. Is that okay? Or do you have...
Whoa, yeah.
Is that a lot? I don't know, if I talk really...
Such a quick revision of basic genetics. Thank you. Okay. I learned some things there. That was good. All right. Should we stop sharing?
Yep.
And bring everyone